Hi, my name is Stephanie Kim from Dr. Stein's lab at Seoul National University. Today, I'll be talking about an ultra-fast structural alignment tool that uses a special kind of structural offsets called 3DI. So in order to annotate functions or to trace back evolution of every kind of protein homology detection plays a key role here. Finding distantly related proteins using sequence alignment alone is challenging since the protein sequence can diverge far. However, protein structures are conserved for a longer period of time, so we utilize structural alignment methods to further enhance the remote homology detection. And ever since the recent breakthrough of AlphaFold2, we anticipate a rapid increase in publicly available structures. However, the current state-of-the-art structural aligners are either not sensitive enough or do not scale well to serve such large structural collection of proteins. So here we propose a structural aligner that is sensitive and fast enough, and that also scales up to billions of structural comparisons. This study consists of two parts. First part is developing a structural alphabet that transforms a protein three-dimensional structure to a one-dimensional sequence, and that will be used later for alignment, which was done by Michael from Max Planck University. The second part is implementing the conversion of protein to a 3DI-based sequence to a new aligner that is faster and more sensitive compared to other aligners. So what is a structural alphabet? Structural alphabets are fragment libraries composed of a relatively small number of fragments that complement each other to form a universal code of local conformations. CLE is one of the many other different structural alphabets where CLE letters correspond to a cluster of combinations of three angles formed by alpha carbon pseudobonds of four contiguous residues, ending up with a total of 17 letters. And even though other structural alphabet has performed quite well in representing a three-dimensional structure, there still remain some limitations. For example, CLE does a great job at representing a backbone conformation of small fragments, but it lacks in representing the long-range residue interaction information. The goal of our 3D alphabet is to better describe parts of the protein tertiary structure. In order to do so, we need to consider not only the backbone conformation, but also the long-range interacting residues. So to define the residue-residue distance, 3DI used a completely free position called virtual center, which is defined by three parameters: angle of the plane containing C alpha, angle of the plane containing C beta, and the distance relative to the backbone. The interaction between the residues is described by the relative orientation of their C alpha backbone fragments, ending up with a total of 9 features containing 7 angles describing the backbone orientation and 2 distances describing the long-range interaction. And these features were clustered to get discrete states, and each residue was assigned to a state by the nearest neighbor, and we ended up with a total of 17 letters. Now that we have generated our own structural alphabets that also describes the long-range residue interactions, we then implemented the transformation to our sequence aligner. The module called Easy Search can search single or multiple queries formatted in PDB or MCIF files against the target database of protein structures. And by going over these 4 inner steps, first the 3DI conversion, second the creating DB database stage, third the pre-filter, and lastly fourth the Smith-Waterman alignment, it outputs a tab-separated alignment resolve files. The pre-filter module computes the similarities between all sequences through a very fast and sensitive k-mer matching stage, followed by an ungap alignment. The alignment module implements a vectorized Smith-Waterman alignment of all sequences that pass a cutoff for the ungap alignment score in the pre-filter module. We made it easier for the users to use with a single command line called Easy Search, and this will produce an alignment of any given input PDB files. So that's the overview of our Easy Search module pipeline. Now, how well does our aligner perform compared to the other tools? So we compared our 3DI, colored in red, with 4 different tools: Structural Alphabet CLE, aligned by Smith-Waterman, colored in blue; 3D Blast, colored in purple; TM Align, colored in yellow; and MM62, our baseline, colored in green. We used SCOP database to compare the performance, which contains a total of over 11,000 proteins. And one of the reasons we chose SCOP is because it is one of the databases that classifies protein by their structure. And SCOP's hierarchy of protein classification goes by family, superfamily, fold, and class in an ascending order. And as you go up the hierarchy, the lower the structure similarity you will get, but they do suggest a probable evolutionary common ancestry. For the runtime comparison, we ran an all-against-all search of over 11,000 proteins in a single core for each tool. And unfortunately, we ended up estimating the runtime for TM Align after running only 10 query proteins against the entire SCOP database because TM Align was too slow compared to the rest. The scatter plot here shows sensitivity quantified as the area under the curve value versus runtime and log scale. and they are separated in 3 SCOP classifications: family, superfamily, and fold. The closer you get to this upper left corner, the better sensitivity and speed you have. And you can see that 3DI dramatically outperformed the rest of the structural alphabet-based tools in speed and in sensitivity for all 3 categories. And our tool was 100,000 times faster than TM-Align with pretty close sensitivity in the family category. We expected that as you go up the hierarchy, the worse alignment it will be, and that is the trend that we see for all of the structural alphabets plus aligner tools, and also for the TM-Align as well. For the sensitivity, we have first ran an all against all alignment of SCOP proteins. Then we calculate the sensitivity for each query by counting up until it finds 5 false positives on the sorted list of alignment scores. From there, we have generated the ROC curve to measure how well it did in picking out the true positives from a pool of false positives when they are sorted by their alignment score. For quantitative measurement, area under the curve value was calculated. Once again, there are 3 different ROC curves for different classifications: family, superfamily, and fold. As you can see, Seq3D outperformed all the tools in all classification with fairly high AOC values, especially in family classification. You can see that 50% of the queries were able to tease out at least approximately 80% of the true positives, while the remaining 2 had approximately 75% sensitivity. In the superfamily category, 50% of the queries were able to pick out at least 40% of true positives, which is a dramatic decrease compared to the family. But when compared to the other 2 structural alphabets, it was able to pick out approximately twice as many than 3D Blast and CLE. Once again, as you go up the hierarchy, the structural similarity is lowered, thus it becomes very difficult for any of the structural alphabets to classify the proteins. Nonetheless, our Seq3D did exceedingly well compared to the other structural alphabet methods. In this study, we successfully developed structural alphabets that contains more structural information by representing not only the backbone of the structure, but also local fragments through its nearest neighbor interactions. And we also developed an aligner that is capable of aligning billions of protein structural alignment for classification. You can use Seq3D by compiling from source provided here in GitHub. Currently, the easy search module is available. And thank you all for listening.